This is Lalita Singh and you are going to study science with me in class 7. Today we are discussing second part of chapter 13, that is called reproduction in plant. In our previous video we have studied about type of reproduction, okay, and we have discussed about asexual reproduction. Understood? And we have also talked about vegetative propagation by natural method. Now today in this video we will discuss about artificial method of vegetative propagation. Actually, vegetative propagation is a very simple and fast and less expensive method to propagate new plant. That is why we commonly use in horticulture and agriculture. The technique used are stem cutting, layering, grafting and tissue culture. So we will discuss about them one by one. The first one is stem cutting. You can see. Okay, in this method, cutting off a healthy young branch of a plant which is having leaves, bud, is planted into the moist soil. What we do? We will cut a healthy branch from the plant. Which type of branch? A branch which is having leaves on them, which is having buds on them. Okay, now we will plant the branch into the soil. The cutted branch develop new roots. They get anchored into the soil, and with the help of root they can absorb the water, and leaves are already present in the stem. So by absorbing water, the leaves can still perform photosynthesis and the plant will now grow into a this branch will grow into a new plant. This method of propagation has been used in chameli, rose, sugarcane and bougainvilleas. Okay, now next is layering. Next method of propagation is layering. Layering is the same as subaerial stem propagation, but what is the difference over here? We in subaerial stem, the branch or the stem of the plant grow on their own, but over here we have interfere. Okay, so in layering, a young branch is bent toward the ground and we cover them with the moist soil, okay, forming a layer over it. After some time, after a few days, the root develop from the nodes of the plant. The branches then cut off from the parent plant. Just see over here, this is a plant, okay, in which we want to perform layering. For that we will pull a branch or a stem of the plant toward the soil. We will cover it with the soil. Okay, after few days, this branch which was covered into the soil, they will grow roots. As the plant new branch is having roots, they are having shoots and leaf, they will perform again photosynthesis and they will become stabilized. And it does means that we can now cut the connection between the parent plant and the daughter plant. Okay, so it is basically done by us. So we are interfering again and again. We have put the branch into the soil, we have cut the branch after the development of root. So that is why it is artificial method of reproduction, that is called a layering, because we are forming a layer over the branch. Next is grafting. Grafting is a common method used in horticulture to develop new variety of ornamental plant and fruit trees. In this method, the bud or cutting with the bud of a plant is called scion. Okay, this is called scion. The upper portion of the plant which is having a bud, okay, is called scion. It is a foreign substance for the main plant. Okay, it is a foreign substance, means it is a part of another plant. This scion is kept over the cutted stem of rooted plant which is known as stalk. Stalk is the one which accepts scion. Okay, stalk is the one with root, whereas scion is what? It is a foreign branch of the plant which has been grafted over here. Okay, now scion and stalk together, they are firmly tied. Okay, like this. Now scion and stalk are firmly tightened up over here. They are tied up. After someday you will found that now these broken part of the branch get fused together, and now they are becoming a single part of the plant. They become a single plant. Okay, the care is to be taken that stalk has an extensive root system under the soil so that it can even support the growth of new branch. Okay, and after some time, the tissue of the stalk and scion join together to form one plant. The stalk supplies the essential nutrient to scion. Means the stalk over here, the plant stalk is having root which is absorbing water and mineral. They supply the water and mineral to the scion, and from scion to other part of the plant. Grafting is a common method to develop new variety of roses, guava, mango, lemon and oranges. Okay, by using this technique, we can add scion of different plants together with the stalk. Okay, stalk is basically the plant which is having a root and which is connected with the ground as well. So we can add several scion to several branches of the plant, and these scions are with different qualities. Like if we are talking about roses are available in multiple color, so we can add the scion of multiple color flower in the same plant. In this way, we can have several color of flower in only one plant depending upon the branch. Okay, every branch will grow, they will form new branches, and each of the branches will produce similar kind of flower. Means if we have connect a rose scion with pink flower, this branch will grow, okay, and form many of the new branches, and we can have pink flower at those branches, whereas the second scion which was added to it was of with red flower. Then it will grow and it will produce red color flower. Okay, all the branches which has been produced by particular red color flower scion, they will produce a red color flower. Means in the same plant you are having two colors flower. One is of red color, whereas second one is of pink color. And of course, the plant has its own color flower. Suppose the plant is with white color roses. Okay, so in the same plant we are having three colors flower: red, white and pink. Understood? Now what is the advantage of grafting? Plant flower and fruits of desired quality can be obtained. Means like if we like a certain species of guava, they are very sweet to taste, this fruit size is also big. So what can we do? We can graft, we can make a scion from that flower to attach it with the stalk of another plant. Okay, we can make a scion of that plant of which fruit we like, and we can attach this scion from the stalk of other plant. In this way, these branches will grow and they will produce the fruits of similar taste to that one which we have tasted. Okay, understood? Now new varieties can be developed. Of course, we are producing more than one kind of flower and fruits in the same plant, so it is also new variety is produced in this process. The third point is plant which cannot be grown by cutting or from the seed can be developed quickly by grafting method. We are simply extracting a stem out of the plant. Okay, and we are making a scion out of it to attach it with the stalk. If we are not able to produce the flower, or the production of flower is kind of very slow, so we can have multiple plants by using this grafting technique in a very short period of time, by just fixing the cutted stem of one plant over the another. The cutted plant of one plant is known as scion, whereas the one which is going to receive those plant is known as stalk. Clear? See over here, this is how we do grafting. We cut the scion and we attach them with the stalk. This way they are attached together, and now after some duration, after some month, they have been, after most probably 30 days, they have been attached like this, and now they have grown up like this. The tissue get fixed over there. Now they do not need any support to grow further on. Okay, and what happened when we have add stalk? When we connect the stalk of pink color flower with scion of orange color flower, this will result. Okay, a particular branch will produce all orange color flower, whereas the rest of the branches will be producing red color flower. Okay, the scion, when they grow, obviously they will form new branches. On all the branches which has been formed by scion, they will produce what? They will produce orange color flower, and all the stalk which was over there, which now grow, okay, they will produce red color flower. You can see this is the perfect example visible over here. The plant of rose has attached with white color scion, red color scion, okay, and purple color scion. And in this way we are having several color of flower in a plant which was originally of yellow color. Clear? You can see over here as well, yellow color flower is attached with the red color flower. Now this look like a bouquet, right? Just because we have been performing grafting with these plants. The next topic is tissue culture. Tissue culture is also known as micropropagation. In this method, we culture the tissue. Now the question arises, what is tissue? Actually, all organism, either it is plant or animal, does not matter. We are all made up of a small unit which is called cell. These cells are considered as structural and functional unit of living organism. Okay, so one type of many cell get collected together to perform a single function or similar kind of function. Okay, they are termed as tissue. So in tissue culture, we are using a tissue. Okay, a small piece of tissue called explant is cut off from the growing tip of the plant. From the tip of the plant, we cut them out. Tip means like see, at the tip of the plant over here, there is a small bud like structure. We have to cut them out because these buds are very, very important, as they are having capacity to let them grow into leaves. They can also form stem, and they can also form root. So they are very, very important. Now the cells of tissue are separated and kept in a nutrient medium, a medium which is having all the component which is required for the cell to grow. Okay, this is called a nutrient medium. You can see these test tubes are having nutrient medium to support the growth of the explant. Understood? So these cells are now separated and kept into nutrient medium. Okay, and the cell of the tissue started to divide rapidly to form an organized mass of the cell, and that is called callus. You can see this mass of the cell which is present in the test tube is known as callus. Okay, the callus is transferred into now another nutrient medium containing plant hormone. These plant hormone are very, very essential because they are guiding the growth of the plant. Okay, they can help in the formation of leaf. They can also help in the formation of stem and roots. So we have transferred the callus. In case of callus, there is no differentiation, only mass of the cell has been formed. But when we have transferred them into a tissue, these tissue into a nutrient medium which is having plant hormone inside it, so then they started to grow into several parts of the plant, like leaf, like stem or roots. You can see in this diagram over here, the plant has grown into root and shoot, as well as leaves are also developed over here. Okay, the cells of callus into nutrient medium divide and differentiated into to form roots, shoots, leaves. Even they grow enough, okay, so that we can now transplant into the ground to have a new plant. Okay, these plantlets are prepared in the test tube. You can see the callus has grown as a very small plant. Over here it is more clear. Okay, This is, see, they have already formed the leaf, they are going to form the roots. The plantlets so formed are grown in pots now filled with soil. Asparagus, chrysanthemum, orchid, many other plants are grown by this method. Now, what is the advantage of this method? Okay, first advantage, vegetative propagation is a rapid, easy and cheap method of plant propagation. Very easily, in very short duration, we can have multiple plants. Okay, by this method, what do we need? We need a apical part of the plant which is carrying bud. That's it. And every stem of the plant is having multiple multiple number of these birds. If birds are present at the base of the leaf in every plant. Okay, so when we want to have that type of tissue to prepare a callus, it is very important to find very easy to find those tissue. Okay, now the new plant are exact copy of parent plant, as we have extracted the tissue from the parent plant with better qualities. Okay, so those tissue has been grown and give rise to a new plant with same character. Means if we have extracted the explant from a mango tree, which next is demerits of vegetative propagation. What are the demerits? First is, by vegetative propagation, plant lose their reproductive power after few generation. Means after passing the few generation, the plant will started to reduce the number of flower they produce. Okay, and in this way their own reproductive capacity will be shortened up or will be reduced. Okay, next is, due to loss of power to fight against diseases, they are prone to infection. As we are using the callus or we are using the several methods to grow a new plant from the previous plant. Okay, so basically they are more prone for infection. They can get infection very easily. Next is, plant become overcrowded at one place and face lack of nutrient. Like if we are performing layering, or if we are performing layering, lots of plants are together growing in a similar area, so it will become crowded and it will be quite competitive to gain water, mineral and sunlight. Okay, so it will affect the growth of plant as well. These are all the benefits and disadvantage of vegetative propagation and the methods of the vegetative propagation. Now we are going to discuss about sexual reproduction. Okay, we are going to talk about sexual reproduction. Sexual reproduction occur as a result of fusion of male and female gamete. Okay, whenever there is a sexual reproduction, two parents are involved. The one is male and the second one is female, and both the parents are producing a special kind of cell which is known as gamete. Okay, next is sexual reproduction. Okay, sexual reproduction occur as a result of fusion of male and female gamete. Male and female gamete means whenever sexual reproduction is taking place, we need to have two parents. One is male and the second one is female. Both of the organism, male and female plants, will provide their own special cell. Okay, and that cell is known as gamete. What is so special about gamete? Gamete are actually haploid cells. Haploid means the chromosome in the gamete are unpaired in condition, whereas in normal body cells the chromosome are paired in condition. Okay, so to perform sexual reproduction, the plant of both male and female type will produce a special kind of cell which is carrying half number of chromosome. Chromosomes are hereditary material which has been taught to you in the previous classes. Okay, so chromosomes are unpaired in case of gamete. Okay, female reproductive part of plant will carry the female gamete and male reproductive part of the plant will carry male gamete. Okay, so to perform sexual reproduction, these both gamete will fuse together to give rise one single cell that will be known as zygote. Okay, these male and female gamete can be collected from the same plant or can be from the different plant of the same species. Okay, the male gamete in plant is called pollen grain. The male gamete in plant is known as pollen grain. You can see over here. These are the male part of the flower, and the small granules which is present over here, okay, is known as pollen grain. These are the male gamete of flower, whereas you can see over here ovule. This is the female gamete of flower, which is known as ova, okay, which is known as ova, central part. Now the flower and its different part, we are going to discuss about them. Firstly, you must have clear idea to perform sexual reproduction. The plant need two type of gamete, one from male and one from other one is from female. The male gamete is termed as pollen grain and the female gamete is termed as ova. Okay, the pollen grain is produced on the male part of the flower and the ovum is produced in the female part of the flower. This is the female part, okay, which is carrying stigma, style, ovary, okay, and these are the male part of the flower which is carrying a filament and anther. Understood? So basically we have studied in class 6 that flower are the reproductive part of the plant, stamen are the male reproductive part, whereas pistil are the female reproductive part of the flower. So once if both of them, stamen and pistil, will be present in a single flower, that type of flower is termed as bisexual flower, because both male and female gametes are present over here. Okay, both male and female gametes are present over here, so this type of flower is known as bisexual flower. But if a flower can only have male part, means they only have stamen, okay, or they are having only pistil, then that type of flower is known as unisexual flower. Uni means one, only one sex part of the flower is present in the flower, then it will be termed as unisexual flower. Okay, and if the flower is having both of the reproductive organ, that is pistil and stamen, then it will be termed as bisexual flower. Okay, both male and female unisexual flower may be present in the same plant, means some of the plant is having either stamen in them, may be possible some of the plant is having pistil in them in the same plant. Okay, some of the flower of the same plant may have pistil, some of them are having stamen. Okay, mustard, rose, petunia is having bisexual flower, means flowers are having both of the parts. Okay, and papaya, cucumber, etc. produce unisexual flower. That is why some of the papaya plant are not able to produce papaya. Okay, why? Because they are producing more of the male flower in comparison to the female flower. That is why the number of papaya has been decreased from the plant. Okay, now stamen, we will talk about stamen. See over here, this is stamen. Stamen is formed of an swollen apical and a tube-like structure. Okay, stamen is formed of a swollen apical part which is called anther. This stamen, this part, swollen part of the male part of the flower is known as anther, or stamen of the flower is known as anther. This is anther. Clear? And a long cylindrical stalk which is present at the base of anther is known as filament. You can see it is known as filament. Understood? Now a filament is helpful in making them attached with the flower, whereas anther is producing the pollen grain, means male gametes are produced over here in this one. Okay, male gametes are produced in anther. When the male gamete get mature, okay, when the male gamete get mature, what happen? This anther burst open and let the pollen grain out into the air. Understood? The pollen grain float in the air and move from one place to another. Understood? Now the next part is pistil. Pistil is the female part of the flower which produce female gamete. Okay, it is also having three parts. The first swollen part is known as stigma. Okay, the first swollen part is known as stigma. The second tube-like structure is known as style, and the third basal region which is swollen is known as ovary. Okay, this ovary is very very important. Why? Because it is producing the female gamete that is called ovule. When the pollen grain fertilize these ova or ovule, this is result into the formation of zygote. After fertilization of female and male gamete, the first cell that is formed is called a zygote, and this zygote will later on develop into the sheath. Okay, in sexual reproduction, zygote formation take place by the fusion of male and female gamete, which result into the formation of a new individual. Okay, now next is activity number 4. To study the activity 4, we need to have a flower which is bisexual in nature. So we have chosen a hibiscus or China rose. You are very much familiar with the flower. Okay, so we are going to study its different part as well as its reproductive parts. You can see this green color structure over here are sepals. Okay, these green colors structure over here are sepals. These are needed for by the flower to protect the bud. Okay, whereas these are corolla or petals which are needed by the flower to attract the insect to provide the flower a beautiful color. So we will remove all these part because we are going to study the reproductive part of the flower, and this is the reproductive part of the flower. You can see this is the reproductive part of the flower. As we know that China rose is a bisexual flower, so this structure carry both male and female part. Okay, now we are going to study about the male part. These are, you can see over here in the picture, these are male reproductive part of the flower. Male gametes are produced over here. Understood? The male part of flower is also known as stamen or androecium. It is having two parts. The upper top region is known as anther, whereas the lower pipe-like structure is known as filament. In case of hibiscus, the filament fuse together to form a tube which surround the style of the female part. Okay, now these are the anther of the flower which is present at the top of filament. Okay, when they get mature, pollen grains are released from here over here. Okay, now we are going to open the tube which is formed by the fusion of filaments. Okay, now after removing this, we can see the inner structure is like something this. Okay. The upper part of it is known as female part or stigma. The middle pipe-like structure is known as style, and then the lower, the swollen part is known as ovary. Okay, this pistil or which is carrying stigma, you can see stigma over here. These stigma are sticky in nature so that they can receive the pollen grain. Okay, whenever the pollen grain are going to brush this part, they will get stick over the stigma. Understood? Now next is, you can see this is the ovary part. So stigma is connected with the ovary with a long tube like structure which is called style. Okay, understood? And this, when we cut open the ovary, we can see the ovules are present in the ovary. When the flower get fertilized or when they receive pollen grain, they fertilize the flower, and then what happen, the ovary of the flower will become fruit, whereas ovule which is residing inside the ovary, they will develop into seed. So this is all about the female and male part of the flower. Next is activity number 5, to study germination of pollen grain. To perform activity number 5 means to study the germination of pollen grain, we need flower, we need slide, we need cover slip, we need sugar solution and a microscope. Okay, so let's start the study or experiment. First, we will extract the anther from a flower. Okay, the anther should be collected from the flower with the help of forceps. Okay, and then it is transferred to the slide. With the help of needle, we will break this anther. to release the pollen, and also we will try to open the pollen grain. Then we will add sugar droplet, okay, sugar syrup which is kept over here. We will add 1 or 2 drop of sugar syrup on the slide where anther is placed. Understood? Now after that the sugar syrup is placed over here, we will leave it for half an hour. Okay, we will leave it for half an hour so that the pollen grain can absorb the sugar and they can start to germinate. Understood, class? After that we will place a cover slip over the slide. Understood? We will place the cover slip over the slide and then we will study it with the help of a microscope. Once when we have put the pollen grain on the slide, this is the picture. Okay, this is the picture that has been that can be seen with the help of microscope. You can see a round spot like structure. These are actually the pollen grain, okay, present on the slide. Now the same picture is present over here as well, but the difference is over here you can see a slight tube like structure emerging out from the pollen grains. Okay, these are called pollen tube. This is all for today's class. In next class we will discuss about mechanism of sexual reproduction. Until then, have a nice day.